Hello, my name is Hartley Carlson, and I'll be starting the presentation off today on our research, interstellar insects, feeding preferences, and dietary composition of Tetramorium caspitum, or pavement ants, under microgravity. The outline for our presentation will go as follows. We're going to start with some background information, talking about why we chose ants and why they're important for building an artificial ecosystem. Then we're going to get into the research questions of our proposal, the experimental design, and lastly we'll finish with our insights and applications for this research. Interplanetary travel and colonization are growing frontiers across various branches of science. Many researchers and organizations investigate what is truly necessary for these explorations and therefore need to develop efficient, practical, and robust ecosystem models for these missions. These models must survive the journey through space conditions, including microgravity and exposure to increased radiation. And in order to maximize the efficiency of artificial ecosystems and save resources during the long missions, it is therefore crucial to investigate the impact of space conditions, especially microgravity, on the growth, survival, and the adaptation of different organisms. Why should we use ants? Ants have been widely considered as prime candidates to send to space due to their high adaptability, accessibility, and environmental benefits. Ant colonies are essential components of ecosystems because they contribute to a high amount of the biomass, and also they act as engineers. They can modify the habitat structure and change the nutrient content of soil, and also increase the density of essential decomposers. Compared to other organisms, they are very easy to contain and transport. They can be in low oxygen conditions and also survive cooling, which makes them awesome organisms to send to the space station and conduct various research. The general question that this experiment will investigate is if microgravity affects ants' feeding preferences and whether or not it will alter their feeding behaviors. We can look at this through specifically these 4 questions. So we can look into the percentage of food consumed, so how much of the food the ants are consuming on Earth versus microgravity. We can also look at the ant mass loss. This is something that is commonly observed in humans, where there's a body mass loss and changes in appetite. We want to see if this is also conserved in ants. We can look at aggressive or cannibalistic behaviors, which is not commonly observed in ants, but it has been seen when they're under stressful conditions. They can elicit cannibalistic behaviors. And lastly, we would like to look at the optimal foraging theory, which this predicts that animals will feed in a way that maximizes the energy intake per unit of time, and ants' feeding preferences could change based on the influence of microgravity for this theory. So they could eat different foods with larger sizes, which would maximize energetic benefits of those.
Now we have our experimental design. Volume 1 is our nest area, where 15 pavement ants will be kept at until their arrival on the ISS. Minimal essential media and water will prevent ants from starvation and dehydration during their transportation and flight until they arrive at the International Space Station. After the arrival, we will ask the astronauts to unclamp clamp A and gently shake the tube. This will give ants access to the nutritional resources located at Volume 2. Several nutritional resources will be glued to the roof moss, which makes the ants' accessibility to them easier. Moss will also provide oxygen to the ants. These nutritional resources were chosen based on previous studies on the Earth. For example, since ants in the presence of gravity prefer sugary solutions combined with amino acids, dried peach was chosen as a source that contained sugar and protein. Sugar crystals were chosen as a primary source of sugar, hemp seed was selected as a primary source of protein, oat was picked as a primary source of carbohydrates, and lastly, peanuts were chosen as a source that contains fat, carbs, and protein. In order to terminate the experiment, we will use formalin, and the reason that we selected formalin is because it will preserve our nutritional resources and our ants' bodies.
Lastly, I'd like to talk about the insights and potential applications that this research will have. First, we would like to build an efficient diet plan, so lay the groundwork for understanding what the most efficient and best composition of certain foods is for these ants. On extended spaceflight, you need to absolutely justify everything you bring, and therefore it's necessary to understand exactly what you need for these ants and what will maximize their efficiency and survivability. We also will study the effects of microgravity on body loss. It is often observed in humans, and this is a phenomena that could be potentially conserved across vastly different organisms. If we observe this in ants, this is a very important feature to know, so then we can look into more ways how to prevent it. We would also like to better understand the potential mechanisms for behavior, so this could be aggressive behavior or cannibalistic behavior. Anything that's induced by microgravity would be important to know if you'd like to send ants in space. Together, all our research is a good starting point for future research that is needed before we can send ants or other similar insects on extended space flights. I'd like to give special thanks to my other group members, Jared and Willow, who could not present with us, as well as our mentor, Dr. Frauwirth, for everything he put into guiding us and developing this research proposal. I also would like to thank Terps in Space and all the other research teams that contributed to this undergrad research program.